Thanks for coming. This was an awesome turnout. So my name is Melissa. I'm the volunteer coordinator here, and I also help with some of the public events. I see a lot of new faces, so sometimes I don't give a little spiel of who we are, but I think I will today. You are sitting at the sort of educational hub of the Great Bay National Estuarine Research Reserve. Has anyone been here before? All right, so lots of hands, but definitely a few that haven't. So we are a national research reserve. We're part of a national system. There are 30 reserves around the country. Every reserve is part federal. So about 70% of our funding comes from NOAA, and then each reserve is about 30% of their funding comes typically from another state agency similar to New Hampshire Fish and Game like us, so like a Fish and Wildlife type agency. The Wells Reserve in Wells, Maine, who's been there? I was just there this morning. Yeah, so that's our closest reserve. They partner with a private trust, so they're a little bit different. But and we also have a friends group, a nonprofit friends group that helps us fill in the holes. And every reserve has 4 core programs: research, land stewardship, coastal training, and education. And so this is kind of part of our broader education program, public lectures. We like to try to get people here, hopefully teach you something new, connect you to Great Bay. And today we have some great speakers who are experts in their field, and we're really excited to have them. This is our last lecture of the winter season. I'm glad it doesn't feel like winter out today, though. So Wynn Watson was a UNH professor for a very long time. He's a lobster expert and also a horseshoe crab expert. And then Chris Chabot is with Plymouth State University. He's been working on the Great Bay Horseshoe Crab program for years. And for the last couple of years, Great Bay NEAR has been partnering with both of them to help out with that program. So today you'll learn all about horseshoe crabs. So thank you.
Thank you. I'm the Wynn Watson part of this duo, and we don't have like this full hour of things to talk about because we want you guys to have plenty of time to ask questions. So feel free at any point, raise your hand so we can address your questions at the right time. You don't have to save your questions till the end, okay? So what we're going to do is give you a little background about horseshoe crabs in general. We're going to tell you what we think are some of the coolest things about horseshoe crabs. And of course, that's going to touch a lot on our own work with horseshoe crabs. And then we're going to talk to you about what we know about the horseshoe crabs that live in Great Bay and their behavior in Great Bay. And we're going to ease into this spawning phenomena that we're getting ready to study in May and June. Okay. We're also going to talk to you about why it's important that we do these surveys, because currently there's some concern about horseshoe crab populations all up and down the eastern seaboard, because for a couple of reasons the numbers have gone down. And so we're going to spend some time on that as well. Okay. So that's kind of the big picture. Okay, so let's go way back. Let's go back 450 million years. Based on the fossil data, they've been here for a long, long time. They clearly are very well adapted to living on this planet. And they're also very adaptable, flexible, because they live everywhere from Mexico up to Maine, right, in estuaries and in the ocean. So they're very hardy animals. And they can also live with very little oxygen. They'll bury in the mud, and they can survive under those conditions as well. So they're very tough animals, and they've been here for a long, long time. There's several different species. You guys are familiar with the ones that live on the east coast of the USA, but there are also some that are over in Asia as well, and that population is also in big trouble. So, as many of you know, from just walking around the beaches in May and June around here, all of a sudden, sometimes you'll just see 100 horseshoe crabs crawling up from the beach. You go down to Chesapeake Bay and stuff, it's crazy out of control. There's so many of these guys. And so for years, they've been used for things as crazy as fertilizer, right? And who cares? There's a bazillion of them, and they're readily available, and they're free, and let's just use them for that kind of stuff, right? And so for a long time, really nobody's been concerned with whether they're going to survive or not. That's going way back. So going back not so far, like when I got my PhD in the 70s and 80s, I became fascinated in these creatures for a number of different reasons. One of the things I studied was their circulatory system, how they get oxygen. You said we have some of these guys lying around?
In that blue cooler.
All right. I'm giving a talk here for a bunch of fifth graders or something like that, a couple of years ago. And, I'm like halfway through the talk, kind of pull out a live worship crowd. It was all over the place. Just had, it's still okay out there, everybody had to hold it. Which is probably the way it should be. And feel free to come up and play this. So, these things down here, right here, these guys, these are the gills. Okay? And they're called book gills because they're like pages of a book. Every flap has a whole bunch of these pages. And they're kind of like the plastic pages of photo books with two layers. And so blood flows through those pages and gets oxygenated as they flap their gills. Okay? So these guys breathe by flapping these gills, circulating blood over them, and the oxygenated blood gets moved by the same flapping movements, pumps the blood up to their heart, which is located all along the top of their carapace. So it's a long tubular heart. And then it enters the heart, and the heart beats and pumps it out. And those two kind of work together. Same way your atrium ventricle kind of blood. The gills kind of flap close, oxygenates the blood, pushed up to the heart, and the heart will expand, goes in the heart, and pumps it out. And then we have a pretty sophisticated circulatory system. It's called an open circulatory system. But it's still pretty good. So they have this heart along here, and they have the gills, then they have these legs. You can see the legs here moving around, and then their mouth is right there in the middle of their legs. And if you put a little bit of food in there, they start chewing like this. Very stereotyped movements. And as we'll talk about later, we feed these guys. As part of an experiment I'll talk about later. And we'll get to this, but this I'm holding it here. This nail, these front two appendages, they look kind of like clocks in like this. And that's what male holds on to female with during the spawning season. Tail can't hurt you, everybody tells you. Pick them up by the tail. It's okay to pick them up by the tail. There's parts of the body that we don't even know. What's that? And then their eyes, right here, they have eyes all over. 2 eyes here, eyes underneath, eyes in their tail. They have eyes all over the place. These 2 eyes, guys want to know they'll rise back in the 60s and 70s by studying using these eyes to figure out how we attack land and so on and so forth. It's been used as a experimental animal for years because it has certain features that make it very accessible for studying basic processes. Any questions about this guy while I'm holding him? All of it. This guy? We got some, like 10. These guys, like lobsters, they'll molt and grow every year and keep going like that. These guys will do the same thing, but after they get to a certain age and reach sexual maturity, once they become adult, they stop molting. Okay, so they don't get bigger and bigger and bigger and bigger. And males stop molting. I think they'll stop molting like one molt short of females. Females are always bigger than males. Okay. Was there another question? Yes. So you're, yes, blood vessels all over, but your lobes connect to the main jewels. Yours is all connected together. Blood does exit and enter from your arterial, but these guys, when it reaches the end of the artery, it's kind of squeezed out into the tissue. So it's open and it's not open.
Given the room is so full, this actually might help a little bit.
I can talk. I know this is weird. This is weird. I'll talk. Should I? Yeah. Better.
It's not usually this full, so you're very popular.
I will try to use it. So this is what it's like to have an out-of-body experience. If anybody wants to come up and play with these afterwards, you're happy to, especially if you're under 20. So anyway, they have this interesting circulatory system. And they're, instead of red blood cells, they have, and hemoglobin, like you have, they have this stuff called hemocyanin. Hemoglobin, as part of the oxygen binding part of the molecule, has iron in it. Right? You've all heard of iron, fluorine, and all that stuff. Right? So your blood is red when it's oxygenated because of the hemoglobin. They have copper, so their blood is blue. And that's a big difference. And we'll get to later, their blood also contains another important substance. Yeah, very valuable.
Great, thanks, Chris Chabot, Plymouth State. So why don't you talk a little bit now about some of the reasons why it's so important for the ecosystem and for we humans as well. The adults and juveniles dig to feed, and they dig especially the muddy tidal. Here's a female and a male that just actually came up May 9th, a couple of years ago, just emerged. She and he probably spent the whole winter underneath and probably 40 feet of water underneath the mud, and they came up to spawn. In addition, with digging to spawn in the upper intertidal, they'll also dig in the muddy intertidal. And by the end of the season, the beginning of the season, those mud flats are really very flat, but by the end of the season, they look like a moonscape. I don't know if you can see all of these pits, but that's basically every single one of those pits is feeding a male or female. So they do something called bioturbation. They churn over the mud and bring nutrients up to the surface for a whole bunch of other organisms and allow them to survive as well. So that's a really important ecosystem service that they provide. In addition, horseshoe crabs lay lots and lots of eggs up and down the eastern seaboard, as when showed you all that you can in areas such as Cape May, New Jersey. There are millions of these animals that come in, and millions and millions of birds that come in and feed on the eggs. Now, don't feel too bad for the horseshoe crabs. The eggs, they lay about 109 tons in one area, and these birds arrive emaciated after nonstop flights of 2,000 miles or more, 40% body loss of their weight, and they fatten up over 2 weeks, and then they fly on to the Arctic where they breed. And what happens is, as these horseshoe crabs dig in the sand and the mud, they turn over more sand and bring eggs to the surface. And these animals basically eat those eggs that are on the surface, lots and lots of them. So without this food for migrating birds like the endangered red knot, it would be very problematic. And we'll talk a little bit more about that later on. So in the last 30 years, Wynn and I have been, 20 years, I guess, 30 years, 20 years or so, we've been studying these animals. And as you, most of you know, as we've talked a little bit about, during the spring, horseshoe crabs mate and spawn. The females come up to the beach, often with the male attached. She digs a nest, she lays her eggs, drags him over, he releases his sperm, and then they go off. And generally spawn 6 or 8 or 10 times more at other beaches for the rest of the 6 to 8 weeks. Males, oftentimes, there's more than one male that's involved in the spawning events, and we'll show you some pictures of that later on. You can see lots here. You can't tell who's who here, however. So more than one male can fertilize eggs, and so you'll see that when a female lays eggs, the male in the back, you might imagine, gets most of the paternity, but actually, one of the, they call satellite males, males that are on the outside, actually have a little bit higher paternity than the male that's attached on the back. So back in the 1990s, 30 years ago, the assumptions dogma about these animals were that they come in to breed and spawn during the spring, and then they go offshore and are tens of miles offshore during the winter. The other dogma was that they only spawn around new and full moon and high tides, only at night. And actually, there's a beautifully illustrated book called Crab Moon that I remember reading to my kids, where they say they have to get up at night to go out and see the crabs spawn. So these are kind of some of the dogma. Lastly, the dogma was, the assumptions were that these guys had circadian rhythms of behavior, which didn't really make a whole lot of sense to us because we knew they spawned according to the tides. Circadian is, circa is about a day. And those are the rhythms that we all had. And horseshoe crabs have them as well, but we didn't really believe that they had circadian rhythms of behavior. So that's where we came along and started to do some work on horseshoe crab behavior.
So basically, Chris told you all those things that we could tell you that we found all those dogmas to be wrong. So we started studying the horseshoe crabs in part by putting tags on hundreds of them, and then asked people to report if they'd seen animals with those tags. We also put, you can see, this is a tag we put on the side of the shell. It's a sonic tag that puts out these pings every so often. And with a hydrophone, we can hear the pings. And more recently, we've had tags like that that'll also transmit depth and activity and things like that. So we can learn a lot about their behavior while they're actually running around in the estuary. And you can see this on the slide slide over here from one of our papers. You can't read the numbers, but basically this horseshoe crab was down here in Little Bay near Newark's in the winter and moved all the way up here past Jackson Lab towards the Discovery Center and in the middle of Great Bay in the spring, stayed there for a while, and then moved back down here in the fall and winter. So this is sort of the typical movement pattern we see for these guys. They don't go all the way out to the ocean. They stay within Great Bay. They typically overwinter down Little Bay in a deeper part of the estuary. And we also found that this movement was somewhat temperature dependent. And I think, let me see. This is a cool video. Should I jump down? Okay, so I was telling you that we could track these guys. Okay, and so this is a video showing the track of one of these horseshoe crabs, a female, and showing how she moves into, oh, maybe your best, showing how she moves into shore to spawn. And this is, again, this is the middle of the night, over about 7 or 8 hours. Oopsie, my bad. That was me.
Yep.
She's right here.
I can,
And you can see she moved over here and right along here. This is Jackson Lab over here, Adams Point, and she did her spawning thing right there. So with data like this, we can quantify this female spawn how many times, right, in a given spawning season. And data like this were also used to show that they spawn during the day high tides and the night high tides. They don't care if it's a new moon or the full moon. They don't do, females don't spawn every high tide. They'll do it like every, what, eighth or something like that. So females don't come to shore at every high tide. I think they have to recover a little bit from laying the eggs and everything else before they spawn again. Males come into shore more often. Yes.
Does the thing tell you how long she's there to know that she actually spawned in that spot?
This is a stupid question. We're gonna ignore it. No. Totally valid question. How do we know she spawned? We don't. So we call these apparent spawning events. Okay, we're not sure that, you know, obviously if she stays for a while, we have confirmed on a number of occasions, you guys, or we will have walk in the beach and we'll make a note. Saw email number 4985 buried and spawning today. And then later on, as we're looking through the videos, we can say, oh yeah, she did spawn. So we did confirm a number of occasions. I don't know many examples of seeing that and having a like she didn't spawn, right? But that's a very good question. Thank you. Anything else? Okay.
Just get it going again.
Okay.
I think it'll keep going.
Right. So, as I suggested earlier, we found that many of these dogwoods weren't true. They don't just spawn on the new and the full moon high tides. They don't just spawn at night, right? And so these are the data here showing that they don't just spawn at night, right? So this makes you wonder if these guys are so synced into the tides, how do they know when it's like that? Right? So either they have a clock that's set on 12.5 hours, or they can sense the change in water depth, right? And it turns out it's both. And that and one reason that the dogma was that they spawn on new and full moons is, if you've ever been down in Florida, the tides are much smaller than they are here. And the only tides that are really big enough, I think, for them to detect in Florida are the new and full moon tides. So that might be part of it. And that's sort of where that for early studies were done. I think it's, this is good.
So when set me up quite nicely to talk about clocks and rhythms. And basically, as we talked about earlier, they have two kinds of rhythms. One is a circadian rhythm of eye sensitivity, and when his graduate students work out with this eye cup, that flashlight every minute or so, and look at the eye sensitivity, and what you see down here in this nice looking graph here, it's not dark, it's just right here, is that at night their eye sensitivity is much higher than during the day. And when we put them into constant darkness, that rhythm persists, so they actually have a circadian clock, just like we all do, that helps to drive this rhythm of eye sensitivity. And what this does is it makes their eyes much more sensitive, almost a millionfold more sensitive at night. So they can see just as well under a full moon at night as during the middle of the day. They also show, and we showed this back in the first publication in 2004, that they have circadile rhythms of behavior, as we talked about a little bit. So we came up with this kind of crazy idea to put horseshoe crabs on running wheels. Put them together with 5-gallon pails. This is the bottom of one, the bottom of the other. They're facing each other, have a nice slot for the tail. And they rotate that, and every time they move around, there's a magnet that trips a switch, and so we know when they're active. And we've had lots of these running wheels in buckets. And we showed that when we put these guys, these horseshoe crabs into constant conditions, they do have those circa tidal rhythms. And in constant water level, there's an internal clock that will drive them to become active about every 12.4 hours. And that's what these data are showing up here, these darker lines. This is activity for 5 minutes. So this is 1, 2 times per day, 3, 4, 5, 6, 7, 8 times over 4 days. That's about circa tidal. You guys all know, a lot of you anyway, that the tides come about twice a day. And that's what we're showing here. So as Wynn mentioned, they can also sense changes in water depth. And that helps them to synchronize with the tides out in the bay. So they have the internal clocks and the ability to sense the tide, to synchronize their activity for spawning and for feeding. So what about other populations and younger stages? So we did a study a few years ago where some graduate student, Tyler Thomas, and I went down to Florida, and we collected animals from the east coast of Florida, Indian River Lagoon, around just south of the Cape Coast, over here St. Joe's Bay, and then of course up here in Great Bay. And the interesting thing about these 3 different populations of horseshoe crabs is that they're exposed to very different tidal frequencies. Those of us who've grown up in this area or know this area really well know that there are 2 tides per day, but other areas don't have 2 tides per day. So believe it or not, you go into the Gulf Coast of Florida, north of Tampa, there's 1 tide per day. That's it, because of primarily the shape of the Gulf here. As well, over here on the East Coast, it's 0 tides per day. They're in the Indian River Lagoon, which has very little water flowing in and out during tidal cycle, so it's essentially a 0 tide population. So we asked the question, what kinds of rhythms would these animals show? And we used a system to
Go back and if you go backwards and come to the slide again.
yeah, that's probably what I'll need to do, yeah.
Go on slide back.
But you can see these guys moving here. These are little tiny juvies. They're very small. They're in very small 612 plates. And they just hatched about 2 weeks ago, and they're moving around. And we videotape them, and then we quantify their movements. And we can record hundreds of these guys at a time, and we do, and we look at what kind of behavioral activity they have, essentially. So this program quantifies the distance moved, and they're divided into what I'm going to show you next, which are called actograms, activity graphs, essentially, showing when they move and what their patterns are. So again, 3 different populations, 3 different tides per day, 2 tides per day, 1 tide per day, 0 tides per day. And we brought them into the lab and show you these data here. Here are Great Bay animals up here. Now, I just want to explain what this actogram shows. These are 4 days worth of data. This is a little bit screwed up, but basically this is 0 to 48, 24 to 48 hours. So on the bottom is time of day, and on the x-axis is day number. So this is day 1, 2, 3, 4, 5, 6, and so forth. It's about 20 days worth of data. And what's important here is not that you completely understand what an actogram is, but the pattern of activity that they show. And you can see here 1 bout, 2 bouts, 3 bouts, 4 bouts for 2 days. That's about 2 bouts per day of activity. That's circa tidal activity, according to this northern East Coast. But if you look down here in the Indian River Lagoon and in St. Joe's Bay, Florida, how many bouts do you see per day? Just 1, right, exactly. So these guys actually have a different kind of a tidal clock, which makes a lot of sense for where they grew up. And we showed very convincingly that it has mostly to do with genetics as opposed to the tides that they're exposed to. Because what we did is we collected eggs from all 3 of these sites, freshly laid eggs that within 20 minutes of them being hatched, put them in buckets, flew them home to Plymouth, New Hampshire, and put them into 3 different conditions, either 0 tide, 1 tide per day, or 2 tides per day. And the conditions that we gave them didn't matter much. It was their genetics that seemed to matter greatly.
does the slope of the graph signify how the tides are changing daily?
It really does, yeah. So this is about a 12.4 hour period. Absolutely, good eye.
Just a curiosity question, so if you move back to the 0 side to 2 side 0, it will not survive there?
Yeah, we wouldn't do that in the field, but we did that in the lab. They did perfectly fine, but they only showed when you take a 1 tide per day animal and you put it into a 2 tide, they're just going to exhibit 1 bout of activity per day. It's an interesting question. Yeah. So point here is that the different populations up and down the seaboard here have different genetics, and as well, we can use these tiny little guys that we have in eggs to really help us answer a lot of important basic biology questions. We in fact we released about 200 today from the winter that hatched last year out in Great Bay. Another question?
Now they make them different species, or they do the at all?
So different populations. They certainly at this point would call them the same species, but give them a million years and we'll see. That is an experiment we're running right now. It's a good question, yeah. So they seem to be the same species, but definitely different populations. So it wouldn't be ethical for us to take animals and transplant them. Now, they do fine likely, but it's not do that. Brings us to the next segment, whether they are on the clock.
No, it's an interesting question. How many subpopulations are there? Right? With the genetic tools we have these days, it's very easy to do the genetics and try to figure that out, how much mixing has occurred. So let's rewind about 30 to 40 years. And people down in Chesapeake Bay area, the Audubon bird people, are noticing there's some issues. The bird populations are declining a little bit. The birds aren't looking good. And so they started raising some warning flags, and the people down there in charge of keeping track of the horse crabs and whatnot for the state started looking into that, into the situation. And basically, the bottom line is that the horseshoe crab populations were declining significantly. And oddly, the first people to notice it were the Audubon people, because nobody was doing horseshoe crab surveys back in those days because there were so many of them, right? And one book that was written on this whole story, The Narrow Edge, by Deborah Kramer, I highly recommended. She came to visit us, talked to us, and she really is a smart woman and has her act together. And she did a very good job of describing this whole situation. And as you can see here, this shows catch per unit effort, right? In these surveys they do from 1990 till 2005. And here's that decline I was telling you about, right? So the numbers were plummeting and people were beginning to get very worried about this. Why were they declining? Well, there's 2 major reasons. One was that some fellows at Woods Hole, Fred Bang amongst them, discovered that there was a substance in their blood that was very sensitive to gram-negative bacteria. So it was kind of like their immune system or your white blood cells. And you could extract this substance. It became called limulus immunocyte lysate, and you could mix it with, before you got a blood transfusion, mix it with a vaccine, whatever. You could test substances that were about to be put into your body at the hospital to see if they were contaminated before you did so. They used to do this by injecting rabbits with these and seeing if the rabbits would respond. So you can imagine how expensive and time-consuming and stuff that was. So this was quite a discovery. But this LAL stuff was very, very sensitive. Okay. And so literally now, everything that goes into your body gets tested with this. And you can imagine during COVID, with all the vaccines that are being produced, really a huge need for this material. They get the material by bleeding these orchard crabs shown here, and they remove about 30% of their blood volume. Now, all that blood you see there is not made of LAL. Mostly that's regular blood full of that blue hemocyanin stuff I told you about, right? And just like when you give blood, if you've ever seen the tube, right, an hour later, there's a little layer of white stuff on the top, that's your white blood cells. That would be like this. So you need a lot of blood to make a little bit of LAM. So every year, every year they bleed, 600,000, 500,000 orchard crabs to get this stuff. And a certain percentage of them die, 20, 30% of them die. They like to bleed females more because females are bigger and have more blood. And then they release them back into the ocean. And maybe more die, we're not sure. Right? And so Chris and I started looking into this. And we would subject orchard crabs to the same bleeding procedure at the Jackson lab. We put these little accelerometers on them that are kind of like, same accelerometers you have in your phones that tell you how many steps you took in a day. And then we would let them run around, and we'd bleed some and we wouldn't bleed some, and so on and so forth. And we found out that after you bled them, it took a week or two for them to recover their same level of activity. They were lethargic for a while afterwards. Okay. And a certain percentage of them died, as you might expect. Right? Fewer of them died in our hands than at the bleeding companies, because when you bleed them, they have to pick them up on a boat, put them on the deck of the boat, then drive them to the factory, let them sit overnight, bleed. It's a rough process, in addition to losing that blood. But we also noticed, we also thought when we did this, we wondered if we released them back into their natural habitat, would they also behave differently, right? Would they recover? And would this affect them during the spawning season so they would spawn less? Right? Because if they're spawning less, this could have a big impact on the population as a whole. So we bled females, we collected them early in the spawning season. We bled some, we didn't bleed others. We put those little tracking devices on them like I showed you earlier. This shows an example of a track from one of these females. And we found that the bled females subsequently mated about half as much as the control females, or expressed apparent mating events half as much as the controls. We did, and Megan Owing, one of my grad students, did this in 2020. Now we repeated it again and got the same data. So this is something to be concerned about. The other way that we lose horseshoe crabs is as bait for whelks. So they capture horseshoe crabs, they cut them in half, they put pieces of horseshoe crab in the bait bag, and they fish for whelks. And we lose on the right is a graph of these are horseshoe crabs harvested and used. So the green is commercial, it's bait landings, and you can see right around now it's averaging about 1 million horseshoe crabs per year for the bait people. Now, all of those guys die. Right? Okay. So you have 1 million that are dying from that. And then this black one shows, the dark green shows harvested for biomedical. And then the black bars are estimated mortalities from that, right? So the combination of these two phenomena, starting, 40 years ago, has led to the demise of a number of horseshoe crabs. Right? And this, as I showed you the data earlier, this is what led to that decline that people started documenting down in the middle of the country. So one of the things we're doing now to try to help is to develop an alternative bait for whelks. Doing this in collaboration with a number of different people, including Liz Fairchild, you might know her, she's a professor at UNH. One of the baits we're using for, there's a couple of different places we get alternatives. One is oyster mussel factories, you know, they collect them all, they bring them in, and they drop a bunch on the floor. We get those. Kind of like the way you used to get pig food at the for a marketplace, because everything they drop on the floor, they had to get rid of it. So there's sort of ways to bivalve, but we also take green crabs that you know are sort of an invasive species now. They're everywhere. We take those and we make them into bait. We also take the excess horseshoe crab blood from factories, the stuff they don't use, the blue stuff, and we mix that in with it, and so on and so forth. We make these little bricks of fake bait. We use that. We had some good success with that. So we're very optimistic that that might eventually some commercial entity will start making that on a big scale, and it'll be cost-effective alternatives, so they don't have to kill horseshoe crabs. A lot of it's like we collaborate with people in Massachusetts, like there's a big fishery, Martha's Vineyard area, right, and then down, just all the way down to South Carolina, kind of thing. I keep meeting the tribe, right? And I had some whelks, some conch soup the other day just to try it. And of course, it wasn't native spring. It wasn't the real thing. I need to try the real thing, but most of it is probably going to Asia, where it's a sort of delicacy.
When is anybody working on trying to get the lab speed the collection process?
No, it's someone escort this woman out. Our interaction with the commercial people is strained, right? And we try to. We want to, like Deborah Kramer and others, they want to visit and see how that works. I want to track a horseshoe crab from the time it's caught to the time it's released. I want to see how we want to learn more about the process, but they're very reluctant to let people in. And then when you do the research, they say you're not following our procedures, and we say we'd like to see your procedures, but they don't let us.
I've also heard there's synthetic that's supposed to be.
Yes, yes, they're working, the synthetic is used in Europe already. But this is like $1 billion industry, so it should be a little reluctance. And it just oddly, they're building a new plant in Cape Cod. Yeah, but it's yeah. So they are doing some things, like some companies in South Carolina are creating big holding ponds. So that maybe they can bleed more than once or something. I saw another hand up here something.
So the horseshoe crab is the sole source of the planet when you tell it out?
It's an awesome question, but the answer I think is yes. Maybe that's one reason they've been around so. Like they just have this really, really, really good immune system. I noticed that years ago I used to, I was talking about it, I worked on their heart. Right, and so we kept on trying to cannulate their hearts so we could measure blood pressure, right? And they just clog up immediately because their immune system to detect that, piece of plastic and just attack it, clog.
You said earlier that they try to chemical innovate it now?
Pardon?
They try chemical innovate it now? Can they create an artificial?
Yes.
Okay.
They have successfully created something that works pretty close to it as well, but it hasn't been received FDA approval.
Curious about the numbers. Is there mandatory reporting from either fishermen or the labs that take the blood? Where do these numbers come from?
It's very flexible. For example, one of the things that you hear about, not again, You hear about how they get them to bleed them, and then they sell them to the bait guys. Um, And most of the rules that they've established some best practices for handling horse crabs. But they're best practices, they're not necessarily rules. You know, like when we go out to fish for lobster, we can't keep any lobster below a certain size. So imagine if the rule was, you know, try not to take those lobsters, that'd be different, you're not gonna get arrested. So, again, it could be better than it is, but again, I think it's evolving very quickly because the fact that, you know, it wasn't long ago when there was a bazillion of these horse crabs and no one cared. But now they're, you know, we see they're a very valuable resource. And when they're, we can't let them decline.
Do they release them to where they're caught?
Habitat, or can you move them around? And
They're supposed to release them where they're caught. They don't necessarily do that. And it could have a big impact, right? For example, like they've tried to transplant horse crabs to the West Coast, like at San Francisco Bay and stuff a number of times. No, they're very synchronized to their environment. And as I show you in Great Bay, you know, this is where I spend the winter. It's good here. This is where I spawn. This is where the little guys grow up. This, you know what I mean? So they've been working on this for millions of years. So then they put them in a different place. Right, you might not know where shore it is and where the good spawning places. So they're supposed to, and maybe they try. Again, I haven't, you know, no one's been able to find out for sure if it should happen.
I think a slide or two earlier, you have a bullet that says something about feeding them after bleeding, increase their mobility.
Yes.
Was there any, did you measure mortality after bleeding?
It's a good point. I don't know if I'm gonna talk, you know, maybe I'll talk about it later. Anyway, I put it off because yes, we have been feeding them after we bleed them because we did this one experiment where we showed that after you bleed them, needless to say, their hemocyanin levels go down, you know, like their hemoglobin, so they're anemic, right? And we found out the ones that were most anemic were the ones that were least active. We also found out the ones that were most anemic before we started were the ones most likely to die. So a lot of their mortality, lethargia, et cetera, was a result of being anemic. So we thought, what if we fed them afterwards on something that would help them replenish the hemocyanin, kind of like when they give you a little cranberry juice stuff, you can put it back, but better than that. And so we started again with things like green crabs and so on. I think I have a picture later. To feed them, we put them on their back and we put food in, right? You know, I showed it, right? And watch them eat, right? So we're we're trying to figure out, okay, how do we do this? You know, how do you keep a horseshoe crab on its back by feeding and holding it? So we do this all the time. It's like when Chris and I invented the running wheel, like, what do you do? We're walking around Home Depot. Those toilet rings, those wax rings. Perfect, right? So you can imagine sending that into the granting agency. We would need 50 toilet rings, right? Okay, use this. So, yeah, we feed them and they would in fact recover their hemocyanin levels faster, and they would in fact be more active than the ones that we didn't feed. So we think probably the best thing for some of these companies to do is to actually collect, and as I said, put them in holding tanks. Maybe you feed them before you bleed them, right? And we found that you don't have to put it in their mouth. You can just throw it in the tank. They'll find it, they'll eat it. So we're also very optimistic about that as a way to help reduce mortality and so on and so forth. Thanks for pointing that out.
She has some.
What's been affecting the populations here in the Great Bay system?
We're gonna show you some data in a bit. Our population's been remarkably consistent. We started doing this in 2012, and we've seen no obvious evidence that the numbers are going down. There's certainly some beaches where they used to spawn, that they don't spawn there because of, whatever's happened physically to the beach. But we don't see any obvious decline in their numbers so far.
Along the small step river at our place. And I mean, 40, 50, 60 years ago, but not recently.
Yeah, the shell with the mold kind of thing. Yeah, I mean, maybe there have been some changes. I was talking to some group, and this young one was telling me about, oh yeah, when I was a kid in Kittery, I used to see them, and now I don't. So yeah, there probably have been some changes, but not anything that we've been able to detect in Great Bay. But I think some things have shifted around because of the shoreline and stuff like that.
Is there any commercial operations in the future? Any questions or mapping?
Good point. Yeah, no, right now, they're not allowed to take bush crabs commercially in Great Bay. And every year at this time, I put on my to-do list, check with the Fish and Game and make sure that they, keep that rule. Right, that's a very good point. And one of the reasons, like states up and down the seacoast are doing these surveys. New Hampshire doesn't have an official survey, right, like other states do. But they pay attention to our data. And one of the reasons our data is good, is because it's a controlled population. Right? So if the population is changing just due to climate change or something else, we would see it as well. But our population has been pretty steady.
I know they're a little bit different species, but is this going on in Europe too?
I don't think there's any in Europe. There are some in Asia. They eat them, are quoted, and they're pretty unregulated. There are people over there who are very concerned, and they're doing their best. But and I wouldn't be surprised if there's some bleeding operations. I'm not familiar with any.
Yeah, they're very threatened over there. So it's very different than here. Free species, but very threatened. That's a good segue into counting our local monitors. That means that's you. Keep going.
Okay, as I just said, most states have these monitoring programs, right? And these are essential for looking at trends. If you're regulating any of these marine organisms, you need to know whether the population is going up or going down. If it's going down, you have to do something about it, right? And this leads to Chris.
So pick it up a little bit here. So why monitor? Some quick reminders we have already talked about. They're very important key foundational species. They're bioturbators. The eggs are really important. There's a couple of fisheries, and there are some population declines in some areas, although not in New Hampshire, thankfully. So additional concerns have been brought up, climate change impacts and effects, and importantly, current loss of spawning beaches in Great Bay. So to address these issues, it's really important to monitor and to basically produce a baseline. So we've been monitoring at several different places around Great Bay for well over a decade, 15 years or so. And here's some data from Scammell and Pickering and Great Bay Farms, Great Bay Discovery Center, Moody Point, and Adams Point over here. And these are the different sites across a number of years. And we don't have to get into too thick into the weeds here of the data, but essentially what we see is that at 12,000 sightings over almost 300 observation days, there are some more observed at certain locations, but there's similar densities overall at these different sites. Now these have been selected by us to be good sites. there are a lot of places around Great Bay where the animals cannot and will not spawn. So what about timing? And here are some data over from 2012 to 2022. And the big peaks here are from last year. And you can see basically it was the 8th or so of May, coming right up, that they started. These are the different colors are the different places. But you can see it starts with some peaks and it kind of ends by early mid-June. Interestingly, when we plot the starting dates from 2019, it was a little bit later. From 2018, a little bit earlier. 2017, right here. 22 here. 2019, 2013, that's 16. But look at this over here. It's very interesting. It's 2012. They actually started much earlier, April 19th. And these are the data, the temperature data from that year. You notice some spikes. So this is the temperature right here. And it climbs and then it goes way up. This is, we had a bunch of animals tagged in the bay. And you notice that as the temperature climbs, they start to move up higher in the estuary. And then it cools off a little bit and they don't move. And then it heats up and they move again. So temperature is really key, we're convinced, in terms of these migratory activity patterns. So very quickly, our sex ratio is changing. It seems like it. You'll see about twice as many males per female in 2023 compared to about 10 years ago. We're not sure what this means. This has been seen and much higher ratios in the fish populations, because the fish populations, they prefer the females. So you would expect that ratio to climb, many more males to females. We wouldn't necessarily expect this here, but that is what we're seeing. So the third part of why horseshoe crabs may be declining along the eastern seaboard is possibly habitat loss. There's a lot of monitoring that takes place, as Lynn mentioned, south of us in a bunch of different areas, very important, very economically important down there. And they do beach counts primarily. But if you look around Great Bay, here's some nice, beautiful charts up here on the side. Most of this is marshland, not beaches. And so it stands to reason that marshland may be important for these guys as well. So we developed new monitoring protocols to include marsh sites. We have been developing sediment coring to look at egg density. Not going to have a chance to talk about it too much here, but this is one of our sites. And believe it or not, we see lots of animals at a couple of these sites here. They are very heavily spawning in these areas. So we need to think about marsh sites as well. We just published a paper this year showing female presence. This is from colleagues of mine of ours down in South Carolina and Connecticut. And you see female presence in the marsh and in the beach. And you also see lots and lots of eggs in the marsh and in the beach as well. These are a couple of undergrads that helped with this publication. So marshes are important as well. But do the eggs develop in here? Because we've been operating under an assumption for a while that the marsh area, that mud, that stinky, smelly mud is pretty anoxic, and it is. And so we reasoned when they laid the eggs, they probably, if they were to lay eggs there, they would probably die because not enough oxygen getting to the eggs. But as it turns out, and this should be coming out this year as well, they develop perfectly well in beaches in blue, and marsh are in orange, and very similar at the 1 week stage or at the 3.2 week stage. So marshes seem to be as good as beaches. Importance of marshes, it's limited beach space, less than 1% in Great Bay. And we have documented over the time we've been counting these animals that some of the Great Bay beaches that used to be great for horseshoe crabs are gone. And so we've been planting hobos or temperature monitors in these marshes. And when you look at marsh temperatures in blue versus beach temperatures in red, you can generally see that the marsh temperatures seem to be a little warmer. And maybe this is another reason for horseshoe crabs to spawn in the marshes. warmer temperatures allow them to develop more quickly, get into the water column more quickly, and potentially survive better as well. That's at least our current hypothesis. It looks pretty good so far. So as Great Bay changes, water level changes, are there going to be beach changes? Some of Great Bay beaches are gone at this point. So some ongoing work. We're very delighted with having a lot of help with 50 or 60 of our closest volunteers, some of whom are here, to continue counting to try to provide a baseline to look to see how these animals survive and spawn into the future. We submitted a Fish and Wildlife grant proposal. We're hoping that this is going to be funded to help identify more salt marshes, essentially, and to look at the conditions in those marshes and see why they are good for eggs, and to see whether or not migratory shorebirds actually preferentially go to marsh sites where they breed versus where they don't. In other words, are the migratory birds eating the eggs here as well? Have we had an impact? Yes. Yes. Our survey data has shown that horseshoe crabs made at all tides, moons, and so forth. So states like Massachusetts have changed their protocols when they are doing their assessments. Our data has also shown that bleeding has an impact on the behavior. So some states like Massachusetts have just very recently, a month ago, banned horseshoe crab harvesting during the spawning season. We're very, very happy to have seen that. And we have some very promising data concerning alternative well baits. And hopefully this will soon eliminate the need of horseshoe crabs to bait.
With the most recent protocol change in Massachusetts, have you ever thought about collaborating with other colleges in Massachusetts? Because I went to Salem State and a lot of their biology courses run along the marshes, and we found plenty of horseshoe crabs on campus.
Nice, we're always looking for additional collaborators. So if you have a name, pass it on to us. So
And might be able to dig up some.
Fantastic.
But in terms of the protocols, Massachusetts, up till now, they only do their surveys on 5 days around the new moon and the full moon. So they may have been missing peaks.
They probably have them, because I know a lot of our times during the seasons, and it'd be like we just had a decrease with funding for our biology department, so they closed down part of the campus that would work for funding data.
Yeah, so if you look at this full moon, new moon looks like a peak there, but full moon looks like a trough there. So, and that's what we see basically through the different years, is that sometimes they line up with newer full moons, but oftentimes they don't. All right, the hopes. We hope that the medical community will embrace alternatives to LAL, the new biotech formulation. Looks like it works very well, as Lynn said. They're using it in Europe. And the reason we're not using it here is because other companies are very conservative when it comes to changing over their billions of dollars worth of products. We hope that companies like Associates of Woods Hole, come on, there we go. Come on. Will adopt our suggestion for treating horseshoe crabs better. That every New England state will conduct annual surveys, and that horseshoe crabs will no longer be allowed to be used for bait. And we continue to develop sustainable and cost-effective research plans. You've heard most of them, but we're going to continue to work on all these areas. And one of our new efforts is to look at the effects of some environmental toxins, such as PFAS. We couldn't have done this without a whole lot of help. Many thanks to there's a lag here, sorry. To all of these people that are shown, especially dozens of volunteers, many of you out there. Great Bay Air folks here, fantastic people, and funding and support from all of these places. So thanks very much.